here at the Gardens, and we're hoping to create a new tradition. My name is Erin Deely, and I'm the Director of Recruitment and Engagement for the College of Science at the University of Arizona. And one of the roles that I have in the college is to do outreach into the community, so it's one of my favorite. And it's true outreach. We get off campus and we bring the talent and the research to you. So, and we actually ask our top researchers that are really capable and adept at translating their science into very digestible and for everyone of all ages to understand and enjoy. So we also are very aware that we have a highly educated and professional community in Tucson, so we get the whole range. Come on in, have a seat wherever you like. We're really delighted to have this community partnership with Tucson Botanical Gardens. Actually, Ashley Peterson, who I'll introduce in just a moment, and our Associate Dean, Elliot Chu, had a conversation, and they've just evolved into, let's offer this as a fifth venue where we offer these science cafes. So if you didn't notice the brochures on the back table, we do have a brochure of all 5 venues, all the dates, times, talks, and each series has a theme. And so I'm assuming you're here because you're interested in the theme, and I'll touch on that a little bit. We're really excited. This is the first night, first time for this venue. Yeah, so it's great. Yeah, we're excited. And then I do want to mention, in addition to the brochures on the back, on the back table, those are just for your pleasure. I mean, we have a lot of public outreach facilities. How many of you been to the Mirror Lab by chance on campus? If not, you definitely want to take one of those brochures. I'll mention during Science City, which is part of the Festival of Books each year, those tours are abbreviated but free. So there's a fee-based one, but then I could go on and on. But we also have Biosphere 2, so if you're not familiar with that, about an hour from here, an incredible biome. 7 biomes of glass, basically. And then we have, any of you been to Flanders Science Center? Yeah. Okay, great. It's great to take families too, and Spooktacular is coming up in October, so over the weekend they get student science clubs doing incredible demos for all ages, and then not to mention the cool dome shows. If you haven't seen the new planetarium, it's a 3D dome projection system. It's fabulous. Yeah, and then have you been to the Laboratory of Tree Ring Research? Yeah. Phenomenal, and they upgraded that 3 years ago. It's home place. So those brochures are back there. Also, you'll find an evaluation form, and that is, we want your feedback on tonight's presentation. We share that information with our speakers. Our speaker tonight is very experienced at giving public talks. So, however, everyone likes to get feedback. Since this is a new series as well, in the comments section, if you would share with us the thoughts of what you'd like to see in this series. I mean, obviously we have to be doing that kind of research within the College of Science or at the university, but give us some ideas of topics that you'd like to hear, and we'll put together a series around that. So most often the ideas from participants fuel what we try to put together. Obviously we tied the theme of this series with the gardens. And actually, I'm going to invite Ashley up. She wants to welcome you to the Gardens and give you some updates about what's happening here.
Thanks, Erin. Thanks, everybody, for coming. I just wanted to say what an honor it is to host the U of A Science Cafes here at the Gardens. It meshes perfectly with our mission, which is to connect people to nature and plants through art, science, history, and culture. And what a great service to the community, and how important is science education today and every day and in the future. So we're really, really happy to host. Thank you so much for being here. I wanted to also let you know that the cafe is open until 8, and so is the garden. So please stroll, enjoy a cool-ish summer evening. The last thing I want to say is I'm going to change the rules on you for the October, November, and December science cafes. So when you pick up a brochure, it's going to say that they're free admission 4:30 to 8. Well, in October we're opening, and I just told Erin about this today. So we're really excited to have a nighttime exhibition of larger-than-life lanterns in the shapes of plants and animals created by Michael Wells, who's a local artist. So we have special event fees that go along with that. However, we are still inviting all of you who attend a Science Cafe to stroll the gardens, enjoy that exhibit after the cafe. So don't come at 4:30, come right at 6, and then stay and have dinner and enjoy the gardens afterward. We're closed from 4:30 to 5:30. Maybe that was confusing, so go to our website after today and then write it down, and we'll see you in October. Yeah, thanks.
Yeah, I want to mention, I'm sure Ashley did, and I updated our website with that information. In the back is also the newest newsletter from Tucson Botanical Gardens, so by all means, help yourself to that. We have a lot of classes that might interest you, not to mention the exhibits. In the Science Cafe brochure as well, you might notice we have an iPad. I have a wonderful volunteer in the audience who is projecting this presentation on Facebook Live, so we can reach an even broader audience. So in the brochure for Science Cafe, it'll tell you the links. If you can't come to one of the talks, you want to see it live while it's happening, or you want to click on it at a more convenient time. So we post those on our website, and we have a YouTube channel. And so all those links are available in that brochure as well. And that's true for all of our Science Cafe venues. So be sure if you want to check those out and you're not able to personally attend. Just as a quick questionnaire, how many of you have been to the College of Science lecture series on campus? Okay, those happen in the spring, and we'll be bringing those brochures out for you. We're not here to ask you for money or anything. We just want to send you information if you like this kind of programming. So this is a true partnership, no exchange of money. It's just a community partnership, two good entities working together. And also on that evaluation at the very bottom, if you'd like to give us your name and email, then we'll send you announcements for the Science Cafe's lecture series. That's it. You know, we're not going to solicit for anything. We also are really curious, you'll notice if you're a College of Science alum, you know, we'd love to know if we have alums in the audience. So we won't send you anything or solicit, but for us to be able to count you as an alum, we just need your name and then your email. So we won't send you anything, we just need your name. If you want to be identified as an alum for the College of Science, that'd be great. Well, let me explain the format just a little bit. Tonight's our first night, so a few more announcements than usual. However, I'll give you a brief intro to our speaker. Then they'll give a wonderful presentation. And then because we have a nice small venue, we're just gonna have the presenter repeat questions. We'll take questions from the audience, and then he'll just repeat those questions back and do his best. And we'll make sure that we cut that off at some point if it just goes and goes and goes. So usually I'll just give an alert of the last question. And then depending on Rob's time, if a few of you have maybe a one-on-one question at the back, he'll take some of those. And then, of course, enjoy the gardens. All right, so let me go ahead and introduce our speaker. So just to remind you, the series is the diversity of life, origins, perils, and conservation. And I would like to just launch right in. You can read what that means, but we'll talk a little bit about next month's presentation as well when we conclude the questions. But let me just tell you a little bit about Robichaux, Dr. Robichaux. He earned his master's and his PhD degrees in botany at UC Davis, and he joined the University of Arizona in the Department of Ecology and Evolutionary Biology in 1986. I'm also an alum of that department, so I've got a soft spot for it. I'd also like to say we're the birthplace of this discipline, which is pretty amazing. And then Rob was named a University Distinguished Professor in 2001. And I'm sure you won't be surprised after you hear him talk. And his work, or research, seeks to reverse extinctions tied in Hawaii. And in collaboration with close colleagues, he focuses on the reintroduction of critically endangered native plants and on the link to landscape restoration at large scales. He is the founder and president of the Board of Trustees of the nonprofit Hawaiian Silversword Foundation, and he's president of the board of directors of the nonprofit Tree Mountain Alliance Foundation. We always like to ask our speakers to share a little fun fact about themselves. So for 6 years, Rob hosted a monthly Latin dance social on the UVA campus. And he can still be found around town DJing those dance socials. Without further ado, there you have it, Rob.
So it's delightful to be here this evening, delightful to be with you. I hope I'll get to meet most of you, if not all of you, after the talk. It's also delightful to participate in the Science Cafe series, especially this inaugural series at Tucson Botanical Garden. And for that, I'd especially like to do a call out of sincere thanks to Erin from the College of Science and Ashley from Tucson Botanical Garden and all of the other staff with whom they work that make this possible to bring what we do to you and to share that with you. So tonight I'm going to sort of take a tour of some of our work in the Hawaiian Islands, share with you up to the most recent insights that we've gleaned from that. It's a little bit of a good news, bad news, but then good news discussion tonight. I welcome any and all questions, including at the end, but also if you have a particular question where you're not quite sure about something, please feel free to just ask during the middle of the talk. I'm happy to answer it or to indicate where I'm going to be answering it going forward, so don't be shy at all. So, what's the context for the discussion tonight? What's the backdrop? The backdrop is adaptive radiation, kind of a fancy term, but we all have heard about the Galapagos finches, Darwin's finches, etc. What do we mean by adaptive radiation, which is likely one of the most spectacular processes in all of organismal evolution? In adaptive radiation, a single colonizing ancestor gives rise to an array of descendant species rapidly, where those descendant species differ greatly in the habitats in which they grow, so they differ ecologically, and they differ in their forms, whether for plants, we're talking about vegetative form or reproductive form. And you'll hear me refer to that as phenotypic variation, so variation in how they appear. So we get this rapid diversification resulting in spectacular ecological and phenotypic variation. Prominent examples of adaptive radiation are found on many oceanic islands, but nowhere are they as spectacular as in the Hawaiian archipelago, where conditions appear to favor unusually rapid rates and high rates of evolutionary innovation. And I'll explain more what I mean by that as we go through. Those conditions in Hawaii include the isolation of the archipelago. It's the most isolated major landmass on the planet. The great variation in physical environments, I'll illustrate that as we go through, and the distinctive geological origin from a so-called stationary hotspot beneath the surface of the Pacific tectonic plate, meaning a vent that's going to give rise to islands, and I'll show you more about the consequences of that in a little bit. Among plants worldwide, the premier example of this phenomenon of adaptive radiation is known as the Hawaiian silversword lineage or silversword alliance. Alliance sounds like something you can join, so we don't use that term as much. It's in the sunflower family, the Asteraceae, so a common family with which most of us are familiar. But these plants are extraordinary in the diversity of habitats in which they occur and the diversity of their forms. And let me illustrate that with one of just a few detailed data slides that I'll use, but I'll work through them. These are the places, especially if you have questions, to let me know. In this figure, all we've done is plot the lower and upper elevational range of each of the 33 species in the silversword lineage. This is what inspires us as biologists when we visit the Hawaiian archipelago, because this is evolutionary diversification writ large. And what I mean by that is species in this lineage grow in virtually every terrestrial habitat in Hawaii except coastal strand. They grow from near sea level to over 12,500 feet, almost 4,000 meters in elevation, reflecting the great variation in physical environments that I mentioned a few moments ago. So this is really extraordinary ecological diversification. In addition, species in the silversword lineage exhibit variation in form. They exhibit phenotypic variation affecting virtually every aspect of vegetative and reproductive form. There are trees, there are vines, there are big shrubs, small shrubs, cushion-like shrubs, rosette shrubs, You name it, virtually every kind of known plant form, major plant form, is evident among the species of the silversword lineage. And I'm going to illustrate the exceptional phenotypic diversity with two examples. The first of these is the famous Haleakala silversword. We know it by its scientific name, Argyrophyllum sandwichins. Here it is in full breeding plumage, so to speak. This is a stunning plant. These rosettes can grow for 40 to 90 years, getting to be about 1 meter in diameter. And at some point in their lifetime, they flower. They only flower once, like some of our agaves. We call that monocarpic habit. And then they die. When they reproduce, they generate this massive compound inflorescence or flowering stalk that consists of up to 600 individual sunflower-like heads, each of which may have 500 to 600 individual flowers. So an exceptional reproductive capability. Interestingly, this species, which grows at high elevations on Haleakala Volcano on East Maui, grows together with, or sympatrically with, something that looks radically different. This is Dubaudium inzicii. It's a diffusely branched shrub. Its leaves aren't silvery. They're not covered by the dense layer of hairs that give silverswords their name. It's got these small green leaves. You can see that here. And the flowering stalk looks radically different, right? It's only got about a dozen heads. The heads are small. They might have 15 individual flowers. And yet these two species that co-occur are very closely related. In fact, they hybridize at Haleakala, amazingly. And here's the contrast. So you see what I mean by exceptional divergence in form, sort of captured in these photos. So that's part of adaptive radiation. Substantial ecological and phenotypic diversity. But the big question is, how do we understand their relatedness? How do we discern whether they share a common ancestor? Well, nowadays, the best way to do it is to sequence DNA, to extract DNA from the organisms, sequence it, compare it, including to other kinds of plants, and use techniques to assess the evolutionary history. So this has been done by Bruce Baldwin when he was in Arizona, at the University of Arizona. He's now at Berkeley. So these are all of the known species, given by their scientific name on the right, that are extant now. And this evolutionary tree goes back in time from right to left. And what you see from this reconstruction of the evolutionary tree or phylogeny of the silversword lineage is that different branches occur. As you go back in time, you get this converging to common ancestors, and ultimately you get to this point. This is the single common ancestor that has given rise over time to the entirety of the Silversword Alliance. So we know from DNA data that species in the silversword lineage share a very large number of mutations that differentiate them from the nearest relatives, which turn out to be in continental North America. So that's pretty cool. And this fancy term just means that we are highly confident in this reconstruction. We have 100% confidence in this because of these large numbers of mutations. So it's compelling evidence for shared ancestry. So that's why we say that the Silversword Alliance is the world's premier example of adaptive radiation in plants. There is a lot of interesting information in this that we won't talk about tonight, but I do want to point out one thing. It's possible, using some statistical algorithms, to actually date this, to get an estimate, a reliable estimate, of the age of the most recent common ancestor. Bruce Baldwin and another colleague at Arizona, Michael Sanderson, did that, and they estimate the age of that most recent common ancestor to be about 5.2 million years, plus or minus some variation. The importance of that, as you'll see in a few moments, is that that corresponds fairly closely to the oldest current high island in the archipelago. So what's the inference from that? It suggests that this diversification has been rapid in geological time. 5.2 million years sounds like a long time to us, but it's just the blink of an eye geologically. And their analysis gave us very strong inference for the rapidity of this diversification. And that it occurred within the context of the current high islands. The Hawaiian archipelago actually extends all the way out to Midway, about 1,500 more miles beyond the oldest island of Kauai. But we know that the diversification of the Silversword Alliance is rapid. Another piece of information, and I'm just going to summarize it for you, but in the question and answers, I'd be delighted to show you the graphs to illustrate it for anybody who might be interested. Another very important inference from genetic analyses is that this rapid diversification in form has actually been accompanied by an accelerated rate of evolution of genes that choreograph development. This is really extraordinary. This was one of the earliest demonstrations documenting how this came to be. By analyzing DNA, by exploring patterns of evolution in DNA from these organisms, much as is done with human evolution now, we're actually able to very precisely measure rates of sequence evolution, rates of accumulation of new mutations, especially mutations that alter the encoded proteins of the DNA. And what we found when we did that with the Silversword lineage is that there's a substantially accelerated rate of evolution, specifically in genes that regulate development, choreograph development. Humans have them, plants have them. In humans, they have a fancy name, Hox genes, genes in very early embryonic development that actually choreograph the expression of many, many, many other genes. And it turns out that one of the keys to the exceptional diversification of the Silversword lineage, both in its extent and its rate, is this accelerated rate of regulatory gene evolution. I mention that just to emphasize that studying these lineages undergoing adaptive radiation in Hawaii actually gives us a window into the evolution of life's diversity writ large. Because if we look back through time, the history of life on Earth actually involves many episodes of these adaptive radiations. The origin and diversification of flowering plants, key example. The diversification of primates, the diversification of insects. In many instances, you see these sort of novelties arise, and then very rapid, sort of explosive diversification in the early history of these major lineages. The cool thing about Hawaii is we can study it in real time, which is really awesome. Yes?
So, you may be going to cover this in a few minutes. I thought I caught that you said the common ancestor for the Silversword was originated in the continental United States.
Yes, that's right.
So what is the hypothesis or the theory of how that, I mean, volcanic islands are sterile, right?
Yes.
So possibly through the air?
How do you get that? It's a great question. And how do we even know what the ancestor is? DNA sequencing actually allows us to resolve that. In the case of the Silversword lineage, it's fascinating because DNA analyses suggest to us that tarweeds, which some of you may know from California, these sort of nondescript plants, that green sticky things that are in the grasslands of California when they're golden, i.e. brown during the summertime, those are the closest relatives of the Silversword lineage. And we know that there was hybridization between two of them and duplication of the entire genome in the ancestor of the Silversword lineage. So how in the world did it get there? We don't know for sure, but the seeds, the fruits of these plants stick to birds' feathers, and so the most likely scenario is it was a migratory bird or a vagrant bird that got them to
Okay,
Hawaii.
thank you.
Yes?
Are you able to map out like geological changes and how it might have the cause of that adaptation?
That's a great question. I'm going to answer that. I'm going to do a little bit of a tangent to answer that particular question when I have the age sequence of the archipelago on in a few minutes. So remind me if I just get so pumped up, I'm just blowing right past it. So this is cool. This is really the good news. Now we get to the not so good news, but I promise we'll get back to good news. Because the Hawaiian archipelago is really this magical laboratory of evolution. Absolutely stunning in the diversity of its organisms. Sadly, well, before I get to that, let me mention one other lineage along those lines. This is a so-called Lobeliad lineage. The second of the exceptional examples of adaptive radiation that we see just in plants in the Hawaiian archipelago. In this case in particular, the diversity of flowers is really quite extraordinary, but there's also diversity in the form of the plants, as we'll see. So the silversword lineage and the Lobeliad lineage are really the two examples of adaptive radiation that textbooks use, that most people refer to in plants when they're talking about this phenomenon of adaptive radiation, which I remind you is probably the most spectacular process in organismal evolution. Hawaii also, sadly, well illustrates the challenge confronting the biotas, the floras and faunas of all oceanic islands, all oceanic archipelagos worldwide, particularly from the invasion of non-native species where those invasions are human-mediated, either deliberately or by accident. In Hawaii, for example, there are no native terrestrial mammals except a single species of bat. And yet, Hawaii now has feral goats, feral sheep, feral pigs, feral cattle, and wild sheep and deer, introduced primarily for hunting. And they've had dramatically negative impacts on the native flora. In addition, there are alien rodents, mostly mice and rats, introduced on ships, typically accidentally, and it turns out those mice and rats prey on fruits and seeds and chew on the stems of young seedlings. In addition, there are alien insects. Hawaii has no native social insects, but now it has yellow jacket wasp, for example, and many species of ants that are predatory, especially on the native insect fauna, including insect pollinators of the native plants. Finally, there are the alien plants or weeds that have escaped from gardens in some cases, or were introduced to sort of deal with erosion problems, but they've exploded in numbers, and in a number of instances become ecosystem transforming weeds, where they literally displace all or most of the native plant species. So collectively, those impacts result in this sobering statistic. Hawaii accounts for less than 0.2% of the total US land area. It's just dinky in terms of land area, yet is home to more than half of all federally listed endangered flowering plants. I mean, it's just staggering in terms of the disproportion. The silversword and lobeliad lineages illustrate this. More than 20% of the species in the silversword lineage and more than 40% in the lobeliad lineage are endangered. I want to focus on two of those initially, the so-called Kaʻū silversword and Pele lobeliad. And to do that, I'm going to take us to what is the youngest island of the archipelago. Remember that I mentioned a little while ago that the archipelago originates from a stationary hotspot, which is actually located in about this region. That hotspot's periodically active, producing volcanoes. That hotspot is 18,000 feet below sea level, so if it's active for a sufficiently long period of time, the volcanoes emerge above sea level, creating islands. Then the movement of the Pacific plate rafts those islands to the northwest, resulting in this really distinctive age sequence to the archipelago, such that the island of Hawaii itself is only 500,000 years old, and the islands become progressively older until you get to the oldest of the current high islands, Kauai, which is about 5.1 million years old, which corresponds quite closely to the age of the most recent common ancestor of the silversword lineage that I mentioned a while ago. In answer to your question, we actually can use that evolutionary tree and date how many dispersal events from the older islands to progressively younger islands accompanied the adaptive radiation of the silversword lineage. And what's quite striking is that it's actually a series of mini adaptive radiations, MINI, where they get to a new island, they explode again. They get to a new island, they explode again in terms of diversification. So that's what makes them in whole so exceptional. It is really cool. So we're going to focus for the rest of the evening on this island. I show it here, just blown up, to emphasize that in particular, I'm going to talk about this roughly million-acre, 450,000 hectare area of these three volcanoes on the Big Island, Kilauea, Mauna Loa, and Hawai'i, cooperatively managed, as I'll mention a little bit later. And in particular, I'm going to focus on this area, shown in this topographic map here in multicolors. So this is this area of the Big Island of Hawaii expanded to show Kilauea Volcano, which has been in the news a lot for some reason the last few months, and the eastern slopes of Mauna Loa. These are the youngest and most geologically active areas of the archipelago, and likely the most evolutionarily dynamic parts of the archipelago. So this will be the area that we'll focus on, and in the next slide, I'm actually standing right near that blue one to look up at Mauna Loa Volcano. So we're standing at about 6,000 feet, looking to the summit, which is just below 14,000 feet, up this shield volcano. You can make out different lava flows from the different colors. This is actually the southwest rift zone on the skyline. This is the northeast rift zone. And much of what I'm going to talk about is between those two rift zones on Mauna Loa and on adjacent Kilauea Volcano. This picture is actually taken from Hawaii Volcanoes National Park. It's actually taken from an area of the park that was added to the park. Previously, it was private land, only in 2003. And it turns out, across this landscape historically, there used to be tens of thousands of Kaʻū silverswords, at least tens of thousands, based on the historical sightings and notes and records. We'll never know the exact abundance, but what we do know is by the late 1990s, they had been reduced to a single population that was known. And it was in this area that the park acquired in 2003. And it contains 380 plants. So they went from the tens of thousands to fewer than 400. Why? Primarily because of browsing by alien ungulates that eliminated them from this landscape. So once the park acquired this land area, they gave us permission to go into this population and begin to work with it. What's a Kaʻū silversword look like? They're stunning. Again, at maturity, they can be a meter across. And in this case, the largest individuals will produce a compound flowering stalk that in total will exceed 9 feet, 3 meters from ground to tip. How do we know that? A good friend of mine is about 6'6", and his fingers are just at 9 feet when we measured him. And when he reached up, he couldn't reach the heads at the top of the flowering stalk. So really stunning. Like the Haleakala silversword, they typically flower once in their lifetime and then die, but have many, many flowers on the many heads in terms of reproductive output. We went into this population, and actually many of the plants were quite old but small. They had been browsed but survived. So we actually retrieved a significant number of them, took them back to greenhouse facilities at the Volcano Rare Plant Facility, realizing that they were not, in many cases, likely to reach reproductive maturity in the field anymore. And we produced what we call the managed breeding population. And to our absolute delight, once we put them in cultivation, like the greenhouses out here, they grew rapidly with water and fertilizer, flowered within 2 to 3 years. and many of them flowered at once, which enabled us to do controlled crossing. It's not complicated. We would just go into the population, collect pollen with in these little cups. You can see it here, it's like pure gold to me. It's like awesome. There are like tens of thousands of pollen grains waiting for something to happen. And what happens? We just get these blush brushes. I didn't know what the hell a blush brush was. And I said, I need something to do pollinating. So I described what we were trying to do. And I said, if it's adjustable, that's great. They said, here it is. I said, whoa, it's silver. That's like perfect.
Yeah.
So why is it perfect? Because you can match the tip of the blush brush, which is designed to deposit a dry substance on a moist surface, like powder on a cheek. We use it to deposit pollen on the receptive stigmas, the receptive female parts of the flowers. We can go in and do highly controlled crossing. In this case, we actually bulk collect pollen from all of the plants that are flowering and mix it. It turns out that Kaʻū silverswords, like many species in the silversword lineage, cannot self-mate. They have a genetic system that precludes self-mating. So we can actually take pollen from multiple other fathers, apply it to the flowers on a given mother, thereby maximizing the genetic diversity of offspring produced by each mother. Then we can collect the seeds and grow the seedlings in large numbers, almost like production agriculture for endangered plants. The cool thing is we can track the pedigree of each. We know with certainty, 100% certainty, who the mother is. And we know the group of likely fathers. That means we can very precisely govern how many seedlings are planted out from each mother. And we can balance that. And I'll come back to that point in a minute. So we grow them for about 9 months at the Volcano Rare Plant Facility, tracking the maternal lineage of each, the pedigree of each. We're working on these lava flows, and it turns out that soil is very patchy. So we actually go down and buy surveyors' pin flags, go out and identify areas with at least about 6 inches of soil, preferably more. Then we get friends together, go out and plant the seedlings into this area. The pin flags also allow us to sort of design the distribution of seedlings across the landscape. We take those seedlings in, it's remote, so we take them in these action packers where we mix across mothers within each action packer, guaranteeing that as the seedlings are planted out into the park, they're planted in such a way that they're likely not siblings nearby. So they're likely to cross-mate more readily. And we ensure that no mother in the entire reintroduced population is represented by more than 2.5% of the total seedlings. So we balance. The reason we do that is it maximizes the opportunity for adaptive evolution going forward. And we can talk more about why that is after the talk. So if it works, then they look like this after a few years. They go from about this big when we outplant them to about this big in a couple of years if we're really lucky. And then if it really works, we see thousands of them in the landscape. This reintroduction effort is in Kaʻū, near that blue one. So it's in the Kaʻū section of Hawaii Volcanoes National Park, and that'll become clearer in a few minutes. So this was cool. We said, oh, this is awesome. But as we were doing this, we said, they're thought to be extinct from the rest of the historic known range. We said, what's the chance we can find something if we explore? So we spent a lot of time exploring, including in the so-called Kapapala region, a big broad area in the central part of the historic range. This is lava covered by this shrubland vegetation. Very difficult to walk through. You always have to look down because there are big crevices in these lava flows, and you can fall 10 feet or 500 feet if you're not careful. So as we're going through, we're looking for plants maybe about 1 foot to 2 feet in diameter, not finding anything. Days of searching. And then by total fluke, we were coming up and over a little ridge. I went to step now, I looked down and I thought, what the is that? Got down on my hands and knees. This isn't a 3-foot mechanical pencil. That's just a regular mechanical pencil. This is about 2 inches in diameter, 2.5 inches in diameter. But it's an old plant. Survived browsing, but was growing in this almost bonsai form, kind of tucked under vegetation. So we said, whoa, we got the wrong search image. We flew back in by helicopter into this remote area on several more trips. Literally would scour on our hands and knees. We'd often find a dead rosette, and then there were 3 or 4 of us. We just scour in concentric, ever-increasing circles. We eventually found 150 of these, and we retrieved almost all of them. We called a friend who has a helicopter who flew them back for us right to the rare plant facility. The person who runs that facility, Pele Moriyasu, absolutely stunningly talented horticulturist, got almost all of them to survive. They flowered, same drill. We did managed breeding, grew large numbers of seedlings, and outplanted them in a different part of the national park because we're dealing with a different source population. So just to go back to this figure, the first reintroduction effort that I talked about was down here. The second reintroduction effort is up here in a part of the park known as Kipukukulaleo. So those names now show up. This is Kaʻū, where we started. This is Kipukukulaleo that I was just talking about. How many seedlings did we outplant? Over 10,000 in Kaʻū from a total of 73 founders. In Kipukukulaleo, we outplanted more than 11,000, in this case from 96 maternal founders. Yes?
Well, so how do you control the browsers in these areas?
Great question, and I'll get to that.
Okay, good.
It's awesome. I love it when it anticipates. Yes? Pardon?
The bonsai form that you have to find.
Yes.
Once you transplant it.
Once we transplant them into the greenhouse, give them water, even though they're old, and the old aspect, I like old, I am. When they're old like that, we think that triggers more rapid flowering. They grew to large size. So those things went from 2.5 inches to 2 feet in a year and a half. It was really amazing. So we outplanted more than 21,000. It's the largest known reintroduction effort with an endangered plant on any oceanic archipelago, which is pretty cool, but that's not the end of it. It can't be the end of it for the reason you asked that I'm going to answer in a minute, and for another reason that I'll get to in a moment. Because the acid test is whether they survive and especially whether they reproduce. And not only that they flower, but that seedlings recruit from them. And we've been doing this long enough, starting in 2000, that we're already seeing flowering in the reintroduced populations, and more importantly, we're seeing seedling recruitment. How much? As of summer 2016, we counted just under 3,000 seedlings in the Kaʻū reintroduced population. These are seedlings coming from natural recruitment in the reintroduced population. In Kipukukulaleo, where fewer plants have flowered, it's just under 600 seedlings, but some of them are getting to be quite sizable now. So this is really the acid test, almost. The true acid test will be that these grow to reproductive size, flower, and recruit, and the population continues to increase. I'm going to switch to a second species. I mentioned the Pele Lobeliad a little while ago. Stunning in its flowers. These flowers are about that large, if you can see my hands, and they produce copious nectar. That's a nectar droplet because these flowers were pollinated historically by a large native honey creeper, now extinct, although there are extant honey creepers that we hope will be able to serve as pollinators. So this flower is really just spectacular. This is the Pele Lobeliad, Clementia peliana. The picture that I show you that we took in 2007 is in a greenhouse. And at that time, that was the last known individual of that species on the planet. That's how dire the circumstances are. As of the late 1990s, Clementia peliana, the Pele Lobeliad, was considered to be extinct in the wild. The last known individuals died. By chance, Pele Moriyasu got access to an old seed collection and got one seedling to germinate and grew it for 7 to 8 years and got it to flower. Stunning, just amazing, but incredibly sobering. I mean, it's beautiful. And then you realize that's it on the entire planet. So we said, well, what's the chance if we do some exploring, maybe there's more out there. So in August of 2007, we went into this remote area of Mauna Loa, climbed up this stream bed, went into this area. It was actually a really miserable day because there was a hurricane coming, but we thought, let's get out there before the hurricane hits. You know how it is, if you do field work, you're absolutely miserable. You're thinking, okay, we just make it through the day, but then you think, we're so miserable, something good's gonna happen, right? And sure enough, we're out in the middle of nowhere in this montane rainforest. We look up and there it is. This is the Pele Lobeliad growing in the wild. It's growing about 15 feet up, epiphytically, in the canopy of another tree. Probably the most thrilling moment of my professional life. I almost levitated up to where the plant was. It was so cool. We did intensive searching. We found 5 more. That doesn't sound like a heck of a lot. So 7 in total, but 7 is a lot better than 1. They're growing only epiphytically because ungulates have killed them on the ground. In fact, the understory of this lower elevation forest is invaded by weeds. So it's not good for recruitment of seedlings. And it turns out it's at sufficiently low elevations, about 3,200 feet, that it's in the mosquito zone. Mosquitoes are not native to Hawaii. They were introduced to Hawaii. Mosquitoes carry non-native bird diseases, avian malaria, avian pox. Where you find mosquitoes, you find virtually no native birds in Hawaii. So, sadly in this area, there are virtually no native birds to pollinate. So even though we found the plants, we weren't finding much fruit production the first couple of years. So 2 colleagues, Kaylee Ibio and Jamie Anoka, with whom I work, said, well, maybe there's some trick we can use. So they explored the literature for tropical fruit trees for air layering in Hawaii and said, why don't we adapt an air layering technique to these plants? Never been tried before. And they said, how the hell do we get up to them? So Kaʻū turns out to be a rock climber. So he figured out ways to rope up into the canopy. We then put Jamie on the ropes because she's skilled. She's also a horticulturist. She can work with a razor blade even when she's twirling on a climbing rope. It's really amazing. I don't know if you've ever tried that, but we would shoot arrows to put ropes across between 2 canopies, run a rope up in between, and send Jamie up, 20 feet up. And if you've ever tried it, the biggest challenge is just twirling. So Jamie is trying to cut into these shoots just through the outer bark, not killing them, knowing that there are only 6 left in the wild, and if she cuts too deeply, it will kill the shoot, and there are only 5 branches, 5 shoots on that individual plant. But she did it. What does it entail? You cut about that much of a length of the shoot, strip the bark away so you expose the xylem inside, and then you wrap it with sphagnum, and then you seal it with paraffin. Because this is rainforest, so you got to keep it from being too wet. And then you go back about 6 months later and voila. This is the callus that forms upstream toward the leaves from where the shoot has been cut, right? And then the roots form from that callus. These are roots forming on a shoot system 20 feet up. And then we come in and sever that shoot and take it to the greenhouse, to the rare plant facility. And if we're lucky, it takes. And they all took. Jamie got all but one of them to produce successful air layers, and then they flowered. So we were able to breed them. So with that, we were able to generate over 1,000 seedlings that we reintroduced into 2 key sites in the national park. And fortuitously, the 7th and last known founder we've just recently brought into the managed breeding population. So we have seedlings from 6 of the 7 known founders on the planet now represented in these reintroduced populations, and in 2016 they began to flower. The cool thing is the park people have these great wildlife cameras, so we've put up cameras to see if we can catch video. These are motion sensitive cameras, catch video of these native EEV, which are native honey creepers, stunningly beautiful birds with a curved bill, potentially visiting and pollinating these plants, which would be awesome. So that's pretty cool. You asked the question, why would we just plant them out there if there's still ungulates in the landscape? That is a great question. So what do we do first? Fence and kill.
Yeah.
It's not pretty. We fence parts of the landscape and we remove, euphemistically, all of the non-native ungulates and any weeds and try to control the rodents. The national park has built more than 250 kilometers of length of fence, more than 150 miles of fence. What does that fence look like? This fence goes for mile upon mile, and you have to realize every fence post has to be drilled into lava, including across these really rugged so-called lava flows. These park fencing crews and the fencing crews on state and private land, to me, they're my heroes. It's just extraordinary work because they have to carry the drills. And then they manually drill every hole into lava, put the fence posts in, and then build the fences. And they build these fences 2 meters, 6 feet tall. Why? To keep these buggers, which you may think are really cute, these female mouflon, out. And then we exterminate them from inside. It is the only way that we can reintroduce the plants. So the park has built 150 miles of fence, and they monitor it monthly or quarterly for damage to the fence, tree falls, etc. They repair it immediately. If there's breach, if an animal gets in, they have ways of censusing for animal evidence inside, scat, for example, and then they send in a crew and dispatch the animal, which makes it possible then for these reintroduced populations to potentially flourish for the endangered plants. This is the national park, the brown. So they fenced an enormous part of the landscape in the national park, but it turns out we've also done that on state land here, Pu'umaka'ala Natural Area Reserve, and in significant parts of Kaʻū Forest Reserve, both state land adjacent to the national park. And we've done it over 30,000 acres, about 12,500 or so hectares of private land. So it's this partnering between private landowners, the state, and the federal government to protect a vast swath of the landscape by fencing, removing ungulates, controlling weeds, controlling rodents, controlling non-native insects. Massive management effort. But it means that these populations have the potential to expand. So they will not require our intensive management into the future. Yeah?
Do these plants have any cultural significance or any other kind of significance to the people?
Great question. Some species with which we work do. For silverswords in particular, the closest indication of that we have are for silverswords growing on Mauna Kea at very high elevations in areas with adze quarries. There was a special kind of this volcanic rock that they would quarry at very high elevation on Mauna Kea, and they find silversword leaves wrapped in tapa cloth in some of the caves near these adze quarries. But we don't know, they probably partly were used for fire, just for warmth, but we don't have a lot of evidence of historical use beyond that for silverswords, and not for Peliwo Meliad so much, but it was quite rare even probably by the early 1900s. Yes?
I have a question. It's evident, it's apparent that there's a lot of work and time put into reintroducing
Yes.
these plants. Why is it so important to reintroduce the plants?
Why in the world do we work with these plants? A great question. They are stunningly beautiful. They represent the world's premier examples of adaptive radiation. They are teetering on the edge of extinction. They have no known use to people. Why pick these particular plants? Because they are charismatic. Silverswords are the equivalent of white rhinos or Bengal tigers. They capture the imagination. There are many, many species in Hawaii that are endangered that aren't so charismatic. But the charismatic ones carry public interest in the non-charismatic ones. And there has been tremendous public support for reintroducing these species which represent Hawaii to the world. So there is no utility to it beyond the intrinsic beauty and the evolutionary significance and the message of Hawaii to the rest of the world. Yeah.
I would suggest though that a successful reintroduction, okay, of these particular species would then make other things possible. What you learn from this, even though there might not be any significance to this particular or these particular species, could then be used in some other
yes,
cases.
there's definitely a practical implication of learning how to do things at a large scale. I've just got a couple more slides, so we'll just fold this right into question and answer. It's a totally good place to do that. If you had asked me 2 decades ago what the likelihood is that we would be cooperatively managing tens of thousands of acres that were free of tens of thousands of ungulates at the time, I would have said beyond our wildest dreams. If you had said, maybe we can reintroduce a couple hundred silverswords successfully, I would have said, boy, that'd really be a benchmark. We did not plan to reintroduce 20,000 when we started. But success begets success, which is exactly your point. And as this became possible, initially we're working in small parts of the landscape, and then bigger parts of the landscape become accessible, become fenced, the ungulates are removed, opportunities keep arising. And now we're cooperatively managing 450,000 hectares, over 1 million acres under the umbrella of the Three Mountain Alliance, which is a federal state-private partnership. That's over 45% of the Big Island is under cooperative management, with shared goals of controlling alien species, controlling fire, and reintroducing the most severely depleted of Hawaii's native biotic legacy. So in this particular region, we've actually worked with all known silverswords and lobeliads to have occurred there historically. I didn't talk about them much, but we worked with the Waiakea silversword. We've reintroduced over 5,000 seedlings, and then 5 other native lobeliads, several of which have fewer than 20 individuals in their remnant population. Not quite as severe as the Pele lobeliad, but also quite severe. And the numbers vary. Some we've started with more recently. But this is all of them. So we're actually making progress now, pertinent to your point, knowing what we've learned working at a larger scale with the Pele lobeliad, especially the Kaʻū silversword. We've now been able to translate that into all historically known lobeliads and silverswords in that part of the landscape. And those are just the silverswords and lobeliads. There are many, many other species of endangered plants. Well, that brings us back to adaptive radiation, right? Because it's this linking of reintroduction of the plants themselves to landscape restoration at large scale that offers the best hope that those ecological processes and genetic processes that underlie adaptive radiation will be able to continue into the future. Because our long-term goal is not really just to reintroduce a Kaʻū silversword, but to allow the lineages to continue to undergo adaptive radiation going forward. And we think it's this linking of landscape restoration at large scales with intensive initial management for reintroduction of these critically endangered species, including those that get down to 1 or 5, to bring them back into the thousands, that offers the brightest prospects for restoring the silversword lineage, the lobeliad lineage, and the myriad other marvels of adaptive variation that pervade Hawaii's biotic landscape. But what's the future, really? This entails many people. I would be remiss if I didn't indicate the large number of people that have helped just with this work in this particular place. But I would also be remiss if I didn't show you that kids help us. Because kids are the future. We all know that, right? We're going to be gone soon. Kids become our future, and they become the future for these lineages. So we've been able to work in parts of the park, fortuitously, with kids from local schools. And I could go on at length about how cool it is and inspiring it is to work with these kids, even if a lot of them is just an excuse not to be in the classroom for a day, and to be outside. But if you look at the hands, and you look at the eyes, it really tells the story. It is the future. So with that, I think I'll continue to answer questions. We can turn the lights on. I'll try to remember to reiterate the questions. Erin may want to make a couple of announcements before we do that.
Yeah, we want to turn the lights back on. And let me just mention, you're all welcome to stay for the Q&A, and I appreciate all your questions throughout. I just want to announce next month's talk. And then I would encourage all of you, if you would please fill out an evaluation. There's a box, you can drop them there. If you want to bring it back, if you come next time, by all means, you can do that as well. So the talk next Thursday, or next month, it's Thursday, October 4th, same time. I should mention too, we chose to be indoors for this talk, mostly because of the lighting. You know, the sun's not setting yet. But we'll be in that ramada at the very entrance, just to the left of the entrance, probably in the future. So it's an outdoor space with fans and all of that. So we'll make sure you guys know that and have signage. So next month's talk very nicely dovetails with Dr. Robichaux's talk, Plant Diversity, A History of Duplications and Innovations. That's going to be given by Mike Barker. He's an associate professor of ecology and evolutionary biology at the U of A. And I'll just give you a quick synopsis. The opportunity to view the diversity of plant life is one of the main attractions of botanical gardens. Plants display amazing variation in form and function. Much of this variation arose quickly, especially when compared with the diversity of animal life. How and when did plant diversity arise? And in this talk, Dr. Barker will discuss the evolution of plant diversity and our current thinking on how it arose. So kind of really expanding on that initial piece that you shared, and then he'll share work from his research on plant genomics that provides new insights into our understanding of plant diversity. And I'm sure many of you have heard of bioinformatics, right? So that is his expertise, and he's actually the director of the bioinformatics program, which is an undergraduate degree at the U of A because of the advances. So by all means, entertain questions. Yeah.
And let me also mention that there, just to follow, I just want to do a shout out to Mike Barker, who's speaking next month, Katrina DeLugash, who's also in the Ecology and Evolutionary Biology Department, speaking the following month on invasive species. So all of these things are going to interlink. And the fourth and final speaker is Brian Inquist, also in our department, who's going to talk about ecology from cells to ecosystems and take a global perspective, again, syncing with all of the other talks. So it'll make for a really wonderfully intertwined series, and they're all awesome speakers. So I'm biased, of course, because they're in the department I'm in, and I think it's a wonderful department. But I would encourage you to come because it'll be totally cool. So questions on anything? Yeah.
You know, there are just a small group of very tiny native bees in the Hawaiian Islands. Honeybees are recent invasives. Do you see any of these native bees pollinating silverswords? Are they all being mobbed by honeybees?
Great question. So the question is, in essence, what pollinates silverswords? I mentioned that birds pollinate lobeliads. Turns out they can also self-pollinate. So that's a good thing, since they're in some cases down to very few. But historically, they would have been predominantly outcrossed by the birds visiting. For silverswords, it turns out to be this diminutive, gorgeous little yellow-faced bee, native bee, in the genus Hylaeus. And fortuitously, those populations are still healthy in the areas where we're reintroducing them. Why? Because the park and the state have been very good in some of these areas at tracking for ants. And because Hawaii has no native ants, you can bait with formicide. It just kills them. Conservation in Hawaii is a lot about killing. If you're not into killing, it's tough, but you kill ungulates, they're easier. You can shoot them, you can trap them. Ants you can poison because there are no non-target ants, native ants that are impacted. That's the biggest threat, especially Argentine ants, which are predatory. And they've gotten into some areas of Haleakala National Park and they devastate yellow-faced bee populations. There are honeybees that show up in these areas, although their numbers have declined in recent years. Interestingly, in Haleakala, where it's been studied, honeybees, which are non-native, which are very effective foragers, turn out to be not as effective in generating high seed set as the native yellow-faced bees. And we don't know why, but we suspect it's because honeybees load up their, tell me if I get this wrong, their pollen baskets. They visit mostly one flowering plant and then they go offload it. Yellow-faced bees, which are these diminutive little cute things, they fly from head to head and they forage. Interestingly, this is a long answer. That's my problem. I can't answer anything about silverswords quickly. We have now observed that native yellow-faced bee, so that individual flowers of silverswords, the female parts push up through the flower, the anthers, the male parts aren't exposed, but the female parts exert and carry pollen out with them. And yellow-faced bees forage like these are popsicles. And this happens about 8:30 in the morning. So just like clockwork, if you're there, you can see yellow-faced bees show up and start foraging. The problem is if they're honeybees, they're just like these big, impolite things, bullies really, and so they're just, so the yellow-faced bees are kind of SOL, they're out of luck, to put it politely. So what we've noticed is now they show up a little earlier and they actually extend their proboscis down into the flowers to forage for the pollen even before it's presented, which the honeybees don't seem to do. So why are yellow-faced bees still there? Because there are these Diwali species in most of these areas that share the same pollinators and didn't suffer near the population decline. Why? Because they're not rosettes, they're diffusely branched. When the animals browse them, they're almost pruned, but they survive. And we think they maintained healthy, relatively healthy yellow-faced bee populations. So the biggest threat is ants. And yellow jacket wasps were known to forage on them too. But we monitor closely in our reintroduced populations for them. More questions about anything? Latin dance. There's a social this Saturday. I'll be DJing for it. Movement culture down on 9th.
How much time do you spend in Hawaii each year?
How much
time
do I spend?
Yeah.
So it's like a commute. I go over about 5 or 6 times a year. Timed to flowering, fruiting, seed collection, sowing, growing the seedlings in the greenhouse, helping with landscape restoration. A lot going on, but that large group of very close friends and colleagues who are all based on the Big Island. And you see that they include this University of Hawaii Rare Plant Facility, the National Park, this nonprofit organization, this state program, wonderful program called the Plant Extinction Prevention Program, state, private entity. This is a trust, an educational trust, federal and federal. So it's this wonderful mix. Now, sometimes, it's especially common for bird reintroduction efforts, it's really top down, right? You go to planning meetings, they have flip charts. I don't fault them for that. What we discovered with plants is we have non-meetings. We don't have meetings inside a building like this. We actually plant together. we need it. We need the shared labor, the shared expertise, shared resources. Sometimes federal dollars are more abundant, sometimes not so, depending on who's in the executive branch. We won't go there. So, other times tourism is high, so state income is higher. Other times the private trust has been able to help a lot. And so we've been able to sort of leverage all of that. So everybody helps everywhere, which is really cool. So it's really not top down at all. And plants are just different. That's why you're here. I mean, some of you probably watch birds. I watch birds. Some of you love insects. I love insects. But plants, it's like biology is the study of plants and then everything else. And plants, you can give a young child a seedling and entrust that child to plant it and come back and monitor it. It's harder to do with a hummingbird and to come back and monitor. So there's a sense of belonging and ownership to the landscape. Now, the coolest thing, this plant was planted in 2000, the year 2000, by kids from his school. It flowered in 2012, and the flower was the first big flowering year. And I was up there working with them by myself. Well, I don't see people most of the day when I'm in this area. And then I hear this cacophony of noise. I'm thinking, what in the world's going on? And then I realize it's the voices of young people. And I'm thinking, okay. And then they just appear on the horizon, kind of a horde of elementary school kids off the trail, of course, but that's okay. They're coming. Yeah. And they were coming up to see these plants in flower, to draw, to write about them with their teachers. And what they didn't realize is that 12 years earlier, kids from their same school, those same classrooms, and some of the teachers were the same, had actually planted them. So it was really thrilling for them. Some of the kids who planted these now work for the National Park Service. Pretty cool. Yeah?
I have a lot of questions, but I'm curious how many graduate students you worked with on this project, or are there graduate students that are going to take the charge, or what do you think?
That's a good question. To what degree do we integrate the educational mission of the University of Arizona into this? And the answer is surprisingly little. We integrate education in Hawaii in a big way. So we now have, through this Three Mountain Alliance Foundation, a long-standing program called Imi Pono no Ka'aina. It's really in trust of the land. And the folks who run it, Lahela Kamara and her co-workers, for years now have been they interface with most of the elementary, middle, and high schools on the Big Island. They bring kids out. Not just for this, but for planting more common, but somewhat depleted species, not endangered species. So that happens. And then there are other programs for late high school and college. So the University of Hawaii at Hilo. There are a number of programs specifically targeting socioeconomically disadvantaged young people to bring them in to the field of conservation. There's actually a prison on the island, which actually falls within this natural area reserve. It actually used to be partly a prison when we were first working there, but there was a lot of native ecosystem adjacent to the prison buildings. So we worked with inmates. We set up a horticulture program. These were inmates. This was a minimal security prison. And these inmates were soon to be released. And so they participated in this horticulture program and natural resources management program, fence building, et cetera, to learn skills important for natural resource management careers. A couple of students from the university have come, a couple of undergrads and a couple of grad students over the years, but not so much. Why? Because a lot of the work is not hazard-free. And in Hawaii, we can work out all of the logistics of that. It just becomes more complicated from here. So a little bit unfortunate in that regard, but The University of Arizona has been awesome to let me do stuff that really doesn't bring.
Yeah, I'm more interested in just the legacy you're leaving, right? So,
Yeah.
and then the infrastructure you have in
exactly.
place. But I have one other question, and it's kind of a
Yeah.
multifaceted one. So, folks don't often get to know the scientists behind the research, and you've done much more than research. You've obviously done conservation work at
Yes.
a huge scale. Can you just talk a little bit about how you got interested in this field? What motivates you to move past the research the way you have in this conservation movement? And then what do you hope for the future?
Okay, I'll try to make it short so as not to talk so much about myself, but it's a great question. So my background is in basic science. I had the good fortune to go to Hawaii when I graduated from high school, not having grown up there, to surf. It's a marvelous place to surf, and I love the ocean. And I thought this was a great life. I'd just surf for the rest of my life. And my folks said, go to college. It would be better. And I ended up going back to Hawaii to study plants, which is really kind of a total coincidence. So we did basic research, mostly biochemistry and physiology initially, and then genetics. Really fascinating, still fascinating, to learn about this process of adaptive radiation. It's very cool. The challenge that was evident is that the organisms we were studying, which were so stunningly beautiful and held such important insights as we began to learn them, that reflected on life as a whole, as I mentioned, the history of life as a whole, we were watching the populations decline. And so we said, maybe we should at least devote some fraction of our basic scientific work to asking genetic questions that might bear on conservation, which is not an uncommon thing. And we used the same language. We said, we're basic scientists, we'll do this work, and then we'll share it with managers and let them implement it. Failing to understand at the beginning what we now understand, that it's just a continuum, that there is no distinction between basic science and applied science. It's just a continuum. There is no distinction between academicians who do interesting but irrelevant work and managers who do really relevant work. So we tried to bridge all of that, to remove that artificial dichotomy. Likewise, we said there's an artificial dichotomy. You see it in the conservation literature. Those folks who focus on endangered species and those folks who focus on ecosystem restoration, landscape restoration. And the former say, that's cool, but if you don't do intensive management, you're going to lose these things. And sometimes these folks say, you hear it. We're wasting a lot of money through the Endangered Species Act because we're just propping up things that are going to go extinct anyway. And we said, it's an artificial dichotomy. Let's just work across the whole. Let's blur the distinction, because they are both necessary and neither sufficient. And then, the more we did that, it becomes larger. Success begets success. It requires more time. I was far enough along in my career that, in practical terms, I knew the university couldn't fire me if I gave up my research lab and devoted the entirety of my time to on-the-ground action. Why does it work? I'm in a department that values that, because everybody in my department studies organisms that are challenged. There might be other institutions. The University of Arizona is awesome this way, and EEB, the department I'm in, is especially awesome to allow and encourage that. So that allowed me the freedom to transition later in my life from entirely doing basic research to doing a mix to doing on-the-ground action, which is scientific at its core, but it's not basic research. The measure of success is transformed landscapes. It's seeing thousands of these plants come back into the landscape. So it's a different metric, but the University of Arizona allows for that, which is really cool. Really cool. The challenge, to get back to your earlier question, is it's difficult in a basic science department to train graduate students in direct action.
Yeah.
So, oddly, I encourage all students to do their work in basic science. They have a lifetime to do direct action beyond, at whatever point that might be, and at whatever level is appropriate, from 0 to 100%. So, does that answer?
That's a great answer, thank you so much. Any other questions for Dr.
Oh,
Yeah.
Is your restoration effort and conservation efforts only in the national parks area, or do you take it to the rest of the little islands in Hawaii?
Our two nonprofit foundations only focus on Big Island. And initially, we were more closely connected to initiatives on Maui and Oahu and Kauai. And then infrastructure developed on those islands, and things became so big for us that we just centered my attention and those with whom I work on the Big Island. The national park, that million-plus acre area of cooperatively managed lands under this umbrella of the Three Mountain Alliance, when it started, it was 30,000 acres. And that was in the 90s. And then more land area became added to it when the success of it became evident. And then amazingly, this large privately held ranch, Kaʻū Ranch, went on the market. The Nature Conservancy partnered with the National Park under the Bush administration, interestingly. That was acquired and added to the National Park and increased Hawaii Volcanoes National Park by 40%. So that opened a tremendous area. So the core of where we work with this set of species on this part of the island is the National Park. But we work closely with adjacent private and state lands. Why? Because there's complementarity. The fact that the park is fencing and removing ungulates then benefits adjacent lands under different ownership and vice versa. So it gets even more detailed. We, as a nonprofit entity, can actually pay the park fencing crew, as we've done, to do all the fencing on the private land. And it's justified by the park because it protects their boundary. And it builds this bridge between Kamehameha Schools, which is the largest private landowner in Hawaii. It's actually an educational trust left by the last surviving member of the monarchy, Princess Pauahi. Their focus is educating children of Hawaiian ancestry. It's a stellar school system. But they also own a lot of land, and they've now partnered to prioritize conservation, the restoring of Hawaii's native biotic legacy, so intertwined with Hawaiian culture and language and music. So all of this is this synergy. And sometimes there are partnering possibilities where federal dollars can match private dollars, and the more private dollars you have, the more federal money you can get, and vice versa. So it's all been, again, so far beyond where we envisioned it would ever get to in just 20 years. So it's pretty cool.
I imagine you share these results also with colleagues and through papers and publications and conferences. And
Yes, right, absolutely. And all of these entities have their own educational programs and ways of expanding the message.
You guys do, oh, I'm sorry. Go ahead.
So I wanted the third question, what's the future?
Oh, yeah.
What's the future for the plants?
For you.
For
you and this project.
My immediate future is a dance social that I'm DJing on Saturday. And it's salsa
Here we go.
and a Dominican dance bachata, so that's in my near-term future. But all that aside, I am actually retiring from the University of Arizona next April to devote all of my time to working in Hawaii. So my future is this until I die. Now, I hope that's when I'm 100. You may see me with an oxygen bottle and an oxygen mask up on these landscapes, but I think it would be fair to say that silverswords, and to a lesser extent lobeliads, sort of pervade my entire life. They have taken it over in ways, the non-family part of it, in ways that it is a future for me forever, because I'm going to become a silversword for sure. When I grow, maybe I'm going to fall out of the helicopter. That would be okay. Having us dying in place and just becoming silverswords. One way or another, I'm going to become a silversword. And a lobeliad. I don't want to discriminate. Hopefully just not weeds. I don't want to become weeds. So.
I feel lucky. I didn't realize you're retiring next year, so I'm really delighted that we got you for this series.
Yes.
If you all join me in thanking Dr. Robichaux.
That was fun.
I should mention too, the gardens do close at 8 tonight. We extend till 8:30, but if you have some time.